Hello everyone, my name is Ali and today we'll be talking about an example of genetic drift in the fruit fly Drosophila melanogaster. If you enjoyed this video or any of the videos on this channel, feel free to like and subscribe down below. Now, let's start with this example. First of all, we've got 107 populations of Drosophila melanogaster were studied in this experiment. We see here the distribution of the frequencies. The frequencies are that image is going to be present on screen at the moment. Basically, the gene that was studied, or the allele that was studied, was the BW75 allele in 19 consecutive generations. Now, in the first generation, what we see is that the fruit flies had an average allele frequency of about 0.5. After each generation, what the scientists have done was isolate 5 males and 5 females from each population and then allowed them to breed to make the next generation. What we notice is that the progressive normalization of the frequencies, even distribution of the alleles, and the loss of some completely. That means we've got the extinction of some alleles, but for the most part they were getting normalized, that the frequencies were getting stable. Now, it is worth noting that while some were actually going extinct, some were obviously also getting fixed, but to a lower extent. However, what was later happening in the 19th generation is that this effect was amplified. And not the normalization effect, actually the extreme effects, the extremities, where you could either have the fixation of a certain allele, of this particular allele, or the complete loss of it. So this is amplified in generation 19, where most of the populations have either lost the allele or completely or actually fixed it. And some are between these two limits but aren't significant.